What I'm going to be talking about today is mentioned, is introducing the new sliding MS2 on the SciX 8600 ZenoTOF system, and the way in which we're taking advantage of this. And the inspiration for the title here, Back to the Future, you'll see in a little while, but it goes back to some experiments that we had originally proposed really over a decade ago, but were never able to do until the launch of this instrument. So we're very excited now to be doing some of these experiments for the first time. The talk today is going to contain two parts. First, I'm going to provide an overview of the workflows in metabolomics and try to point out where some of the gaps are and some of the rate limiting steps are. And then in part two, I'm going to be discussing how we've deployed the ZenoTOF 8600 system to try to overcome those gaps and improve the metabolomics workflows. So we'll go ahead and start here with the metabolomics workflows. And I do want to start by mentioning one overview concept, and that is when I say the word metabolomics, I know that this word can mean different things to different people, but I'd like to provide a little bit of a visualization of how we think of the chemical universe in my lab. So we divide the chemical universe into macromolecules and into small molecules. The macromolecules are going to be your genomics, your genetic content, your proteins. And on the right, we have small molecules. And I'm going to use the word metabolomics to be referring to all of those small molecules. So it's not just water-soluble metabolites, but when I say metabolomics, what I mean is profiling water-soluble metabolites as well as lipids, as well as chemical exposures. So everything I talk about is going to be broadly applicable to all those categories. So we'll start with just an overview of the workflow here of how we go about performing metabolomics or lipidomics or chemical exposomics. And it starts, there's nothing terribly original here, but it starts with sample preparation. So we want to isolate those small molecules. Then we're going to analyze them by using mass spectrometry. The workflow we're going to be talking about today utilizes LCMS, liquid chromatography mass spectrometry. You can do these metabolomics experiments with other technologies like GCMS or NMR, but LCMS tends to be the preferred platform because of the expansive chemical coverage that you achieve. After you generate the data files are quite large and you can't analyze them manually, so we do rely on software for data processing. And that usually results in several thousand different signals, unique signals. Sometimes in metabolomics, we refer to those as features. And there's a range there because the number of signals that you get does largely depend upon the context of the experiment. After you generate the signals, you don't want to just stop there. There's not a whole heck of a lot you can do with signals alone. You want to be able to structurally identify those signals with a high level of confidence. So take those signals, convert them into IDs. That does require doing MSMS experiments or fragmentation experiments and matching that MSMS data to MSMS data in databases or from authentic chemical standards. Now you can see here that the arrows on the top are color-coded green. The arrow on the bottom is color-coded red, and they're traffic lights, corresponding traffic lights on the right of the slide. And this is to denote that those first series of steps are relatively easy to do. I think most labs now around the world can carry out sample prep, LCMS, feature detection. But what the bottleneck currently is, and what still represents the rate limiting step of untargeted metabolomics, is converting those signals into identifications. Now, just as a little background to indicate how serious of a challenge this is, I wanted to show you some real data. These data are now quite old. We published these over a decade ago, but the situation really hasn't changed that much. And you can see in this scenario, we're looking at human plasma and we're measuring on the order of about 25,000 features. And the number that we can identify is just this small sliver here off to the right. Okay, so the point is you're measuring a lot of signals that you're not able to chemically identify. Now you might say, well, humans are really complex. Maybe that's specific to humans, but we see the exact same thing for much simpler systems. So these are simply E. coli that were analyzed, cultured E. coli cells. And you can see effectively we get the same kind of result. Lots and lots of signals that are measurable, but a very small fraction that we can chemically identify with a high level of confidence. It raises this question is what are these signals here in the red? We're measuring these things, but what are they? Measuring something that you can't identify is not particularly useful in the context of biology. So we'd like to think about how we can move forward and try to identify what these signals are. Now, to that end, we've spent quite a lot of time over the last decade or so, we and many other groups around the world, to try to understand what these signals could be. And in that vein, what I'd like to do on this slide is develop a picture of what we've come to think of as the roadmap of an untargeted metabolomics LC-MS-based data set. And as I say, this isn't just work that we've done, but this is work that builds on the research from labs all over the world. So the first thing we want to do is we want to recognize that the signals that you measure can either be of biological origin or they can be of non-biological origin. And what I mean by that is that there are signals that are derived from contaminants or are derived from artifacts. Sometimes the words contaminants and artifacts are used interchangeably. We prefer not to equivocate the two. We think of contaminants as real molecules that show up in your data set, but those are molecules that are not derived from the sample itself. And what I mean by that is that they're coming from other places, like perhaps impurities in the solvents, perhaps plasticizers in the vials that you're using, but they're not coming from the sample, so they're not biologically relevant. The other source of non-biological signals that we get are artifacts. Artifacts are not real molecules, but they are denoted as peaks in the data processing workflows. These can arise from things like electronic noise, or they can be small deviations in your chromatographic baseline that the peak detection software falsely says are real signals. The biological signals also come in a couple different flavors. We have those biological signals that are redundant, and we have those biological signals that are unique. What I mean by redundant is that one metabolite, lipid, or chemical exposure can show up as many different signals. The main sources of that redundancy, sometimes referred to as degeneracy, are shown here. A compound can show up as an adduct, different salts, sodium, potassium, ammonium, etc. They're naturally occurring isotopes that occur. Metabolites can stick to one another and form oligomers, and metabolites can break into smaller pieces as they enter the mass spectrometer. The unique signals can either be those that are identified. These are going to be your TCA cycle metabolites, your glycolytic intermediates, your amino acids, your fatty acids, long-chain fatty acids, cholesterols, etc. And then we have those that are unidentified. Okay, and these are going to fall in two different categories. Those that are unidentified because they are truly novel molecules. These are really exciting molecules that people have never reported before. They're not in the literature. They're not in the databases. They're not in the textbooks yet. And then we have those that are unidentified because they are chimeric. Now, if this is a word you haven't heard before, we'll spend just a couple of minutes talking about what chimeric means here in a moment. But this is a real impediment to trying to identify signals in untargeted metabolomics. Now, in our experiences, when we tried to annotate all of these different signals in various different kinds of data sets over many years, this is the relative distribution of each of those signals that we're generally seeing. Again, on average, there are exceptions, but you can see that we measure a lot of contaminants and artifacts. There's a lot of redundancy. And when you look at the things that we actually care about, the novel molecules and the known molecules down here, you can see that it is a very small fraction of the total number of signals that you're measuring. But what I'd like to zoom in on here today and spend the rest of the talk discussing are these category of signals that I've boxed here called chimeric. Now, your initial reaction might be, yeah, but it's so small, say compared to artifacts and contaminants up here and these other things. But it actually is quite significant when you look at the size of it compared to the novel and the known signals. So we want to be comparing that that bar graph there, not to the top, but rather to what's underneath it. Okay, so without further ado, what do I mean by chimeric spectra? Well, when you're doing metabolomics, as I alluded to earlier, you have to collect fragmentation data to support structural identifications. And to do that, we collect a precursor in the mass spectrometer and we fragment it. In this particular example here, I'm showing you 3 different precursors and they're all color-coded. So we have some precursors that are blue, some precursors that are green, and some precursors that are red. And what we like to do is isolate just one precursor in the fragmentation cell and then break it apart. And that means that all the fragments we measure in that experiment, that MS experiment, come from the blue precursor. And when we match it to a database, which is MS data that was derived from a pure authentic standard, you can see they match quite well. But what happens when we fail to isolate just a single precursor in the fragmentation cell and instead we isolate 2 or more precursors? Well, in that scenario, what ends up happening is not only we're now measuring fragments from the blue precursor, but we're also measuring fragments from the red precursor. Whenever you have fragments from multiple precursors, we call those MS data chimeric. And you can see the problem is that when you have chimeric data, they do not match the database because, again, the databases were built off authentic standards, pure compounds, so there's no chimeric data in the databases. So let's start our discussion when we talk about a data-independent analysis, or DIA, a technology that was pioneered by SWOF 15 years ago or so now, pioneered by SciEx, called SWOF 15 years ago or so. And what you're doing here is you're using wide MS isolation windows and you're scanning across the entire M/Z space, one after the other. So you do an MS1 scan, and then you do all these MS2 scans that are partitioned across the M/Z space. And you could do this, and this, I know there's a lot of iterations of SWOF and it's evolved and there's all these much more sophisticated ways of doing it, but this is one of the earlier iterations of it. But you can see, generally that works. It takes about 3 seconds to do, and that constitutes one cycle. Now, when you do SWOF, we acknowledge that you're going to get a lot of chimeric spectra because you're using these really wide MS isolation windows of 25 M/Z. But how do we quantify how much MS data is chimeric? And to do that, we're going to use this technique that was first put forward by Rick Dunn's group back in 2017. And essentially what you do is you measure everything in a scan. And then this is the targeted precursor, and this is the co-isolated analytes. And then you take the size of the blue and you divide it by the size of the blue plus the size of the red. So it's essentially like the fraction of blue that you have compared to the total. And when we do that with DIA, that's kind of SWOF workflow that I showed you earlier, using plasma, a 30-minute run with helix separation, what you end up seeing is that the vast majority of what you're measuring is extensively chimeric. Again, not surprising because we're using a 25 Dalton isolation window and a lot of stuff's going to sneak through. You're not going to just get one precursor if you're using a wide isolation window. So this has been, in many ways, a turnoff for the metabolomics community because you've got a lot of chimeric data and there's a lot of computational burden to try to figure out what to do with this. So we've, in many ways, many groups have transitioned to using DDA, data-dependent analysis. And there, instead of using large isolation windows, you're using narrow isolation windows, most commonly 1 M/Z. And you do the same experiment. We use plasma, 30-minute helix separation, again, 1 M/Z. And these are the results that you get. And this is the most important slide I'm going to show you today, because what it demonstrates is how much chimeric data you get, even when you're using a 1 M/Z isolation window and a 30-minute helix separation. Okay, the y-axis here is the amount of MS data, number of MS data spectra. And the x-axis here is the percent of contamination. And you can see that, yeah, there's not a ton that's really contaminated, extensively contaminated, but there is a lot that has chimeric spectra and that are going to contaminate your data. So what do we do? There's a couple ways to solve this. One way is to use variable retention times. This is a technique that you rely on the fact that your precursors are going to have slightly different retention times. And because your precursors have slightly different retention times, your fragments will have slightly different retention times, and you can therefore deconvolute them. And this has been published by several groups, but most notably MS-Dial is employing this technology. There are some challenges associated with using retention time deconvolution. It doesn't always work, and it does depend upon using the right chromatography. There's also ways in which you can do back-end deconvolution, where you have, instead of trying to deconvolute your experimental data, what you can do is you can take the MS data in the databases and you can convolute it to generate your experimental data. So you're not deconvoluting, but you're convoluting the experimental data, your reference data, and reconstructing it. And that's a technique that we published a few years ago. And it turns out that the database-assisted deconvolution and the retention time deconvolution are highly complementary. So sometimes the retention time deconvolution works, sometimes the database-assisted deconvolution works, but you can see that they both have unique scenarios where they work. But the key thing is here, and I'm going to wrap up here by talking about how we've inserted the ZenoTOF 8600 system into this workflow. The key thing here is that although we've seen about half of our spectra are chimeric, and we can solve a lot of those chimeric data by using retention time deconvolution and database-assisted deconvolution, currently there are no tools to solve this portion here, this 21%. And that means these are molecules that we cannot observe, that we can't currently, these are molecules that we can't currently identify. So what do we do with those? How do we solve those? Well, this is the inspiration for the title of the talk. Over 10 years ago, we published this workflow that takes advantage of sliding MS isolation windows. At the time, it was just sort of a cute idea because there were no instruments that could perform it to scale. But essentially, the idea is that when you do MS, this yellow line here represents an MS window, and you can see that we're isolating two precursors and getting red and blue fragments down here in the MS data. If you move that window to the left and you move it to the right, you can see that you get different proportions of the blue and the red precursor. Here you get a lot of blue but little red. Here you get a lot of red and a little blue. So if you can slide your MS window, then what ends up, it gives you another dimension to the MS data that allows you to deconvolute. And so we said if we could do these sliding MS windows, that'd be really cool. We could deconvolute data, but again, there was no instrument that could do this fast enough at scale. But it worked really well, just in our cute little examples. You can see, I'll just flip through these. Methionine worked really well, valine, sphingosines, a couple examples. But again, we couldn't do it to scale. So that all changed with the ZenoTOF 8600 system, as shown here. This is G, Tristan, Kevin, and Matthew. And what I'm going to show you here just very briefly is data from Tristan, who spearheaded the experiments I'm going to show you. So I've already shown you how swath works. The white lines here are the isolation windows. But when you do this new approach, and there's so many cool different ways that you can run the ZenoTOF 8600 system, and I don't have time to go through it all now, but there are just a million different modes that you can do. I think that there's so many opportunities for innovation and creativity with respect to metabolomics workflows. But I'm just going to focus on this one application called ZT-Scan that they've implemented. Use a 5.1 ml/z isolation window, but you're incrementing that window at 0.1 ml/z. So each one of those little white lines there is a different MS scan, and they're largely overlapping. And so essentially what you're doing is what we said we would have loved to do 10 or more years ago, but we couldn't do, and now it's possible. And so when you do the ZT scan and then you do the deconvolution, you're getting MS data that looks just like DDA data, but you're getting it across the entire MS window. Okay, and when we take this and we apply it to human plasma, so this is human plasma, 16-minute helix mode, a 16-minute helix separation in negative mode. When we do DDA, and we're just using one database, the NIST, and not going through extensive cataloging here. So these numbers, just one mode, so keep that in mind. But you can see that when we add the ZT scan in, we're getting double the number of identifications. And then the last slide I have for you is that there are several other benefits to the ZenoTOF 8600 system. Because of the ZenoTrap technology, you do get better sensitivity, which you're going to hear about from others throughout the summit today. This is an example of phenylalanine. You can see the peak here. It's relatively low intensity. When we turn the ZenoTrap on, it goes much higher. And you can also see that in addition to doing CID fragmentation data, it is possible to collect EAD which in some cases is going to be much richer fragmentation data, as you can see here for the PC shown. We're also getting weekly CVs that are less than 4%, which is really outstanding. If you're going to do large studies, that's critical. We heard earlier about the importance of quantitation, and that's really the key to it. So with that, I'll wrap up. Thank you for allowing me to present. Again, thanking Tristan. And I just want to say one closing thought, and that is that when you know, if someone said, what is the perfect MSMS workflow for metabolomics? You might say, well, if I could get MSMS data in 1 Dalton bins across the entire M over Z range, that would be ideal. But as I've shown you, if you do that, 50% of your data are going to be chimeric. And that's going to be a problem because you can't deconvolute all that chimeric data. And so one could argue that you'd actually be better off doing sliding windows where you get lots of MSMS data, but you have an opportunity to deconvolute it. And that's exactly what the ZT scan is and what SciEx has developed with this new instrument. Thanks so much for the opportunity to present here today. And I'm happy to take a question if we have time.